Excellent. Thank you, Greg, and thanks for having me today. This is material that I originally presented for a group of extension-oriented people, but Rebecca Butcher actually suggested that it might be of interest to this audience, so I appreciate you having me along. So what we're talking about mostly this afternoon is how we're exploiting genotype information in our modern genetic evaluation for beef cattle in Australia. But we'll talk about that around this concept of reference populations and how they fit into the evaluation. So what I'd like to cover today is firstly to address what reference populations are. We'll look at the information we're using in the modern genetic, and that is really the genomic evaluation in breed plan, and focus in really on how this information allows us to make more accurate selection decisions, which means drive genetic progress for these important traits more quickly. And then we'll go on to look at some specific projects that included in this reference population work. Those include some industry-funded ones, large research exercises, as well as a project that we ran in a commercial Brahman breeding herd. And it's an interesting little case study on how this can be done in industry. So I think to kick things off, it helps to understand how this notion of reference population is fitting into our genetic evaluation. And as it stood before these ideas came along, we had seed stock producers within all of our breeds recording information on phenotypes. That's just the traits that we evaluate in the genetic evaluation. So they were submitting that information along with pedigree information. And I describe these this way relative to what we're doing in the reference, but compared to that, these are traits that are relatively easy to measure. They're relatively low cost, and as a consequence, we get relatively high volumes of data coming in for these. In recent years, that data has included genomic information, and that's been steadily increasing as the years go by. And both of those sources of information go into our breed plan evaluation to generate the EBVs that people make selection decisions on. The real development around this reference population idea is that we're now able to intensively record data in these reference population projects. We really focus in on economically important traits, and they're traits that are identified via our breeding objective system. It is, kind of goes along with that they're recorded at lower levels. So it's really important we do that recording in the right animals, and that objective is to be in animals that are representative of their entire breeds. All of those animals are genotyped. It goes into the new single-step breed plan, which is just how we include genomic information in that evaluation. And the real outcome is more accurate breeding values. And as I said, more accurate breeding values means more accurate selection and opportunity to make more rapid genetic progress. So with that idea in mind, the real opportunity that this system now opens up is that we can take a young, unrecorded animal, and this is a specific animal that was presented for sale at Brahman Week a few years ago, and he essentially had no EBV information on him until he was genotyped. That information was put into the breed plan genomic evaluation, and as a consequence of the recording that happened in this sector of the system, in the reference population, he was able to get breeding values based purely on genomic information alone. And it's the combination of our understanding of his relationship to the recorded population that allows us to generate this breeding value accuracy, which is really a game changer. If we can take these relatively unknown, undescribed animals and present them to to buyers and breeders with this level of information, it really changes how we can do things. And I think as we talk about these reference populations, it's important to understand that we're doing a lot of work in these projects, but really any animal that has both a genotype and a phenotype is part of the reference. And that's whether they're part of a design project or not. And it applies to all of the traits in the evaluation. And it means that it's really important for any recording in those animals with genotypes be as accurate as possible. You know, by having both genotypes and phenotypes, their information's really kind of supercharged in the evaluation, and making sure that's accurate recording is key. One of the really important questions that we wrestle with as we develop these reference population sort of projects is how many animals do we have to record to achieve a target level of accuracy? And that's a function of both how much accuracy we want to get out and the heritability of the traits involved. And we can't talk about any subject around these reference populations without referring to this graph. It's one that was produced by Australian scientists Ben Hayes and Mike Goddard, and has been used as a guide in this area for many years now. And what it's presenting is how many animals we need in the reference population as a function of how accurate we want the resulting EBVs to be. And that's done, these four lines are for traits of varying levels of heritability, going from the most heritable traits in green to the least heritable traits in red-ish. And this is a completely arbitrary standard that I've set just for this discussion, but if we say we want to achieve breeding values with accuracies of 0.6 or 60%, we can see that line is cutting across these curves. If we select a example heritability of 50%, it's telling us that we need more than 4,000, about 4,500 records in the reference to drive 60% accuracy in unphenotyped animals. 50% heritability is at the higher end of the traits we evaluate in breed plan, but it includes traits like age at puberty and lactation and estrus interval, which are being used up north to describe female reproduction, and which we're examining in the south in more temperate breeds. Mature cow weights also up at that level. If we've got traits that are at the 30% heritability level, and that's more the average for the sort of traits that we're evaluating in breed plan, it includes all of our scan, fat, eye muscle, marbling traits. Most of the weights we take are in that sort of range, as is percent normal sperm, which we'll discuss a bit as we go through this presentation. And I think as we head down the heritability, it's pretty clear that we need, if I just go back, for those 30% heritable traits, the number of records we actually need to drive this level of accuracy is about double. So, about 8,500, 9,000 records. And as we proceed to less heritable traits, traits like days to calving in the breed plan evaluation, the milk trait that we produce an EBV for, a trait describing whether from a single record and a female cycling at mating, all of those are about 10% heritable. And we can see from this that we need lots of records. We get up to 20,000 and have to keep going before we're going to achieve this 60% level of accuracy. An important component of this is that if this is going to work, we need to have some level of relatedness between the animals in the reference and the animals that are being recorded. And I think it's important to point out that it's really a useful, any research done to develop higher heritability traits will allow us to record less animals in the reference. And reference population projects, is important to say, are not progeny tests. We have the goal of improving accuracy in the whole breed in these reference population projects, not just for targeted sire, and it's for traits of the greatest economic value. So what we want to record in the reference is really summed up by our breeding objective, by our what is driving profitability for our system and for our breed. And that this is just an example of the weightings that are put on individual traits in our evaluation and as an output of our breeding objective development system for breed object, and that drives the development of indexes for our different breeds. We can see that for this Hereford index, we've got relatively high weighting on growth, on mature cow traits, on calving ease, certainly reproduction as days to calving, with yield coming into the equation as well. And if we look at similar sorts of things across different breeds, those traits tend to be consistent, not universally the same weightings, but growth disorder, calving ease, marbling, reproduction, mature cow traits, and retail beef yield are all have some level of importance. With that in mind, it's worth looking at how levels of recording are for those traits. And for our growth traits, generally they're pretty good. Our calving and marbling traits are not too bad for the breeds that put importance on those. Reproduction and mature cow traits, in contrast, are relatively lowly recorded, and yield is not well recorded. You know, we've got to go back to the CRC before we see good levels of recording for yield, and it's an area that we're focusing on at the moment. So I think the key traits we want to include in the reference are traits which are economically important, that drive genetic profitability. Those won't be the same for every breed, and even within a breed, different sectors in that breed will focus on different traits. And profit-based breeding objectives, which are an output of developing selection indexes in breed plan, are a really useful resource in developing our understanding of those objectives. So just looking finally at the projects that we are running to do this, and this I'll get through this fairly quickly. We've got industry-funded projects. The Repronomics project in Northern Australia is a really good example of this. It's a large industry, it's an MLA-funded project led by David Johnson, who works here at AgBoo, and it's kind of building on the results we got out of the beef CRC and really driving this intensive recording of female reproduction for northern breeds in the evaluation to describe these really economically important traits. The project involves purebred Brahmans, Sanders and Drought Masters, which are fairly representative of the breeds in Northern Australia. If the next phases get off the ground, it will also include crossbred animals. Size a source from industry. We do that to make sure we're as descriptive of the larger populations across these breeds as we can be, and all animals are genotyped, and that information goes into the evaluation. The Southern Multi-Breed Project is a similar initiative, more focused on temperate breeds, Herefords, Angus, Shorthorns, Charolais, Wagyus, and also Brahmans who have been brought down to produce linkage with the Repronomics project. And again, we're recording those same sort of candidate traits, or really important traits around profitability, which is female reproduction, mature cow body composition, male growth and carcass traits, and again, all animals genotyped, all animals producing information that will go into the reference population. Well, I think we can very quickly look at this Kairu MDC project, which was a little bit different from the really big research projects, and had the goal of generating the same sort of data, but from a commercial stud herd. So the aims were to focus on male reproduction in a Brahman stud breeding herd, to improve the description of those traits, in a representative sample of Brahman animals, and to gain benefit from that recording, not just at the herd level, but across the entire Brahman breed. So the Kairu herd was a prime candidate for this. For people familiar with the Brahman stud industry. Kairu was an evolution of the Tatras Brahman stud. It was quite a large property in the Fitzroy River Basin of Queensland. They were running about 1,000 Brahman cows that we were able to work with in this project, but that was servicing both their own commercial herd as well as selling bulls into the Brahman stud sector and Brahman commercial sector as well. Unfortunately, that stud was dispersed in 2020 and we had to stop working with them after about 5 years, but the value of the project is still circulating through the Brahman industry, and those animals are still present in the population. One of the reasons that this was such a good candidate was because they've been submitting information to Brahman Breedland for many years. They've been involved in our CRC and other research projects, for as long as those have been going on, and they're one of the best recorded Brahman herds in Australia. So the opportunity we were taking in working with a seed stock herd was to describe this percent normal sperm male reproduction trait. It is in all of the large research projects, all of the males are steered and we're getting carcass information from them, which is really important, but it doesn't give us the opportunity to look at male fertility, and we were grabbing the opportunity at Kairu to look at that for the Brahman breed. So we measured percent normal sperm in the young bulls in this herd over 10 years, while still collecting all of the intensive description of female reproduction, that's age of puberty and lactation and estrus interval in young females as well. We designed the project to include links to as much of the research population as we could, and we knew that this was a herd that was well linked to the general Brahman population. So looking at the results just relative to this discussion around reference populations, we'll spend just a little bit of time on this table, and the information that we'll present is for just a group of selected sires, the number of progeny they have in the Kairu project, the number of progeny they have outside of that project, how many herds these bulls we used in outside the project, and really importantly, how they performed for accuracy for this percent normal trait prior to and following the data collection that took place as part of this project. So numbers wise, for this selection of bulls, and for people familiar with the Brahman industry, some of these herds will look familiar. We evaluated, at the top of the list, almost 150 young bulls who are progeny of this Alf Collins Pole Desire bull, but lower numbers as we go down the list, around 10, 20, 30 animals. But interestingly, and very much targeted, those bulls also had progeny out in the greater Brahman industry, from just bulls used only at Kairu to bulls used, in 19 other herds, 18 other herds. And I think the really key information to take from this graph, from this table, is how EBV accuracies change. So it's the accuracy that's not important, not the EBV itself. And for Pole Desire, who is a well-described bull already, he's still increased, almost 20% of the back of the recording that occurred as part of this project. But for all of these other bulls, this was a trait that was not being well described in the evaluation prior to this. And after it, we see significant increases for this Fern Hills bull, increasing from under 20% to almost 60%. For the Ambassador bull, below 15 to 50%. So really useful levels of accuracy gain. And on average, for the size evaluated for this, we achieved an increase in accuracy of around 30%, which takes them from being, in many cases, essentially not described for this trait to being fairly well described. As far as changes in EBVs go, it very much followed what the theory told us, that there may be some changes. Some bulls, we saw increases in their percent normal sperm EBVs. For other bulls, we saw some decreases, but on average, most of these bulls didn't change a great deal. So if we just park this off to the side, a quick summary of this project. There were 32 sires evaluated with progeny outside of Kairu. All up, there were actual progeny of sires evaluated outside of the herd, almost 25,000 progeny in 75 herds, including 2 of our repronomics herds. And on average, that drove an accuracy increase of around 30%. So I think overall, an industry level improvement in percent normal sperm breeding value accuracies and benefits to the entire breed on the back of this recording in this herd. And it was a fairly economical way to drive that improvement in accuracy. And it's a model that I really found was effective. And it creates opportunities for our breeders to make more accurate selection decision on this real important male trait. And at least as importantly, it's a trait that is also descriptive of female reproduction. So it hits both of these really economically important aspects of productivity in the north, and allows breeders to make selection decisions in a more informed way. So I think just to summarise across the whole presentation, it's really important for people to keep in mind that the reference population includes any animals with genotypes and phenotypes, whether they're part of these, big research projects or not. And accuracy of recording is really important in both of those sectors. EBV accuracy, driven by this, is a function of a couple of things, including how accurate we want these EBVs to be, the size of the reference population, the heritability of the traits involved, and really importantly, the relatedness of target animals to the reference. So I think if we're going to undertake this reference population work successfully, it's really important to identify and measure the right traits. And those are traits that are demonstrably of real economic importance. They're drivers of profitability for the breed and for breeders. Reference populations involve intensively recording hard to measure traits, and the need for that to be ongoing is really key for people to understand. Those numbers that we need to achieve accuracy are numbers of current animals in the population. So we can't just measure, 3,000, 5,000 animals and let that, and then stop doing it. It has to be updated in an ongoing way to maintain currency with the population. And certainly good herd level recording and genotyping makes a really genuine contribution to that accuracy. With that, I'll say thank you, Greg. There's a lot of people to include in the acknowledgements, certainly the funders, MLA, the people involved in all of these big research projects, as well as people involved in their own herds doing this recording and genotyping, and certainly the people working in those research herds as well. Thank you, Greg. I'm happy to take questions.